Hello everyone. This section is for History of Microbiology. After learning this section, you should be able to evaluate the importance of the contribution to microbiology made by Hooke, Leeuwenhoek, Pasteur, Koch, and so on. Predict the difficulties that might arise when using Koch's postulates to determine if a microbe causes a disease unique to humans. Let's say the person who firstly described microbes. Robert Hooke is credited with publishing the first drawings of microorganisms in the scientific literature. In 1665, he published a highly detailed drawing of the fungus Mucor in his book Micrographia. Antony van Leeuwenhoek is considered a father of microbiology, as he observed and experimented with microscopic organisms, using simple microscopes of his own design. The development of microscopes was the critical first step in the evolution of microbiology. Beginning in 1673, Leeuwenhoek sent detailed letters describing his discoveries to the Royal Society of London. It is clear from his description that he saw both bacteria and protists. You can see the picture on the right. This is Leeuwenhoek's drawings of bacteria from the human mouse. The development of microscopes was the critical first step in the evolution of microbiology. After Leeuwenhoek's observation of microbes, the development of microbiology essentially languished for the next 200 years, until techniques for isolating and culturing microbes in the laboratory were formulated. Many of techniques began to be developed as scientists grappled with the conflict over the theory of spontaneous generation. The spontaneous generation believed that living organisms could develop from non-living matter. Firstly, the Italian physician Francesco Redi challenged the theory. He proved that meat did not spontaneously generate maggots. However, many researchers did experiments to debate on spontaneous generation. Finally, Louis Pasteur did experiments with swan neck flask. Look at the picture. He placed nutrient solution in flasks, heated their necks in a flame, and drew them out into a variety of curves. The swan neck flasks that he produced in this way had necks open to the atmosphere. Pasteur then boiled the solutions for a few minutes and allowed them to cool. See the picture below, the right one. No growth takes place even though the contents of the flasks were exposed to the air. Pasteur pointed out that growth did not occur because dust and germs had been trapped on the walls of the curved necks. If the necks were broken, growth commenced immediately. So finally, Pasteur conflict over spontaneous generation. It marked the birth of microbiology. as a science. The conclusion of Pasteur's Svanek experiment is microorganisms are not spontaneously generated from inanimated matter, but are produced by other microorganisms. Pasteur had many contributions in the development of microbiology. He demonstrated that all fermentations were due to the activities of specific yeasts and bacteria, and he published several papers on fermentation between 1857 and 1816. He suggested a method for heating the wines to destroy the undesirable microbes. The process is now called pasteurization. This picture shows Louis Pasteur working in his laboratory. And Pasteur developed the first vaccine for rabies and anthrax. Then let's know Robert Koch. The first direct demonstration that bacteria cause disease came from the study of anthrax by the German physician Robert Koch. One of his contributions is the recognition of microbial role in disease. He established the relationship between Bacillus anthracis and anthrax. This picture shows Robert Koch in his laboratory. His criteria for proving the causal relationship between a microorganism and a specific disease are known as Koch's postulates. Koch's postulates. His criteria for proving the causal relationship between a microorganism and a specific disease are known as Koch's postulates. This is the content of Koch's postulates. First, the microorganisms must be present in every case of the disease, but absent from healthy organisms. Second, the suspected microorganisms must be isolated and grown in pure culture. The disease must result when the isolated microorganism is inoculated into a healthy host. The last is the same microorganisms must be isolated again from the diseased host. After completing his anthrax studies, Koch fully outlined his postulates in his work on the cause of tuberculosis. Then let's see clearly Koch's postulates applied to tuberculosis from this picture. On this picture, you can see the postulate on the left and experimentation on the right. The postulate is the microorganism must be present in every case of the disease, but absent from healthy organism. So Koch developed a staining technique to examine human tissue. Mycobacteria tuberculosis could be identified in diseased tissue. You can see it on the picture. The second in the postulate is the suspected microorganisms must be isolated and grown in a pure culture. So Koch grew Mycobacteria tuberculosis in pure culture on coagulated blood serum. The third of the postulate is the same disease must result when the isolated microorganism is inoculated into a healthy host. So Koch injected cells from the pure culture of Mycobacteria tuberculosis into guinea pigs. The guinea pigs subsequently died of tuberculosis. The fourth of the postulate is the same microorganisms must be isolated again from the diseased host. So, Koch isolated Mycobacteria tuberculosis in pure culture on coagulate blood serum from the dead guinea pigs. In 1905, Koch was awarded the Nobel Prize in Physiology or Medicine for his study in Mycobacteria tuberculosis. Koch's postulates were quickly adopted by others and used to connect many diseases to their causative agent. While Koch's postulates are still widely used, their application is at times not feasible. For instance, organisms such as Mycobacterium leprae, the causative agent of leprosy, cannot be isolated in pure culture, and some human diseases are so deadly. For example, the Ebola, hemorrhagic fever. You can see the Ebola virus on the picture. It would be unethical to use human as an experimental organism. If an appropriate animal model does not exist, the postulates cannot be fully met. Koch developed pure culture method by adding agar to solidify the media. Then, separate bacterial colonies developed after the surface of the solidified media had been streaked with the bacterial sample. Another important tool developed in Koch's laboratory was a container for holding solidified media, the petri dish. We also call it plate. It is very popular use today in the lab. Koch also developed fermentation and pasteurization, germ theory of disease, and vaccination. Then let's see vaccination. The ability to culture microbes also played an important role in early immunological studies. Pure studies on the bacteria that causes chicken cholera, Pasteur and Pierre Roux discovered that incubating the cultures for long intervals between transfers resulted in cultures that had lost their ability to cause the disease. These cultures were said to be attenuated. When the chickens were injected with attenuated cultures, they not only remained healthy, but also were able to resist the disease when exposure to the virulent cultures. Pasteur called the attenuated culture a vaccine. Shortly after this, Pasteur and Chamberlain developed an attenuated anthrax vaccine. The picture shows the Institute Pasteur. Culture-based techniques were also applied to the study of microbes in soil and aquatic habitats. Let's see the discovery of microbial effect on organic and inorganic matter. The Russian microbiologist Vinogradski discovered that soil bacteria could oxidize iron, sulfur, and ammonia to obtain energy. And many of these bacteria could incorporate carbon dioxide into organic matter. And he isolated nitrogen-fixing bacteria. Bajornik made fundamental contributions to microbial ecology. He isolated Azotobacter and Rhizobium. They developed the enrichment culture techniques and the use of selective media, which have been of great importance in microbiology. Sir Alexander Fleming discovered the antibiotic penicillin. He had the insight to recognize the significance of the inhibition of bacterial growth in the vicinity of a fungal contaminant. Okay, this is the important events in the development of microbiology. From Leeuwenhoek, we say the development of microscopes was the critical first step in the evolution of the discipline. The development of techniques for isolating microbes in pure culture was another critical step in microbiology's history. Nowadays, molecular genetic techniques and genomic analysis are providing new insights into the lives of microbes. Microbiology today is as diverse as the organisms it studies. It has both basic and applied aspects. The basic aspects are concerned with the biology of microorganisms themselves. The applied aspects are concerned with practical problems such as disease, water and wastewater treatment, food spoilage and food production, and industrial uses of microbes. The basic and applied aspects of microbiology are intertwined. Basic research is often conducted in applied fields, and applications often arise out of basic research. Nowadays, it is the second golden age for microbiology. An important recent development in microbiology is the increasing use of molecular and genomic methods to study microbes and their interactions with other organisms. Today, molecular and genomic analysis have paved the way for understanding microbes as biological systems, such as recombinant DNA technology. It uses restriction enzymes and DNA ligases. The genome sequencing. It is the first step in genomic analysis. Bioinformatics. It manages the genetic information from the DNA sequencing. And metagenomics. It studies the microbes in environment without culturing them. Here are the pictures show some important events in the development of microbiology. Milestones in microbiology are marked in red. Other historical events are in blank, such as in 1876, Bell invents telephone. At the same year, Koch demonstrated that Bacillus anthracis causes anthrax. Okay, that's all for this section. Here is what is for you.